this session, I think. Yes, I am. All right. So today we're going to talk about cell division and differentiation, growth, replication, and that's not what I meant to do. All right. And some of it actually loops back into the material that we've already covered, which is nice. So we're actually building upon the material that we already went through one time. And I have a session ID on here if you want to log into Turning Point to do some of the questions, because I'm going to add some review questions to this session as well, just to start you along the path of thinking about things, putting it together. I'm sitting in front of the screen. All right, so okay. And so here you have the study slides for the lecture, so you can go over that material. And here the next slide is just the lecture objectives. All right, so I have a question. I have a few questions to start you off here, so to review, that are not on the PowerPoint that you have. And you can play along at home. So when you study the name and structure of a cell, what are you studying? Which field are you studying? You can click all that apply. Although I think I forgot to click multiple responses. So you could probably only pick one because I don't remember doing that, right? You can only pick one? Yeah. All right, well, pick the best choice you have. All right, I'm going to close the poll. 5, 4, 3, 2, 1. All right, so half the people said anatomy, and that is true. And also the microstructure could also, you could make an argument that it's also histology, but we really haven't gotten into histology, so I don't blame you for not thinking that right up front. But anatomy, microanatomy being a subset of that. That's part of like where we all started this adventure. So the next question here, which picture demonstrates the anatomic position? So we have palms down here. We have arms downward. We have palms up. So which one is the anatomic position? Way back when in the first session. All right, I'm going to close the poll. 5, 4, 3, 2, 1. All right, 100% got it right. Good job. I even tried to trick you up, but I tried to find some really good ones that look like they might be legit, but I didn't trip anybody on that. All right, how about this question? Which anatomic plane is in this figure? All right, I'm going to close the poll. 5, 4, 3, 2, 1. Okay, so we had 63 percentile sagittal. So this is stuff that you forget the little details when you haven't reviewed it in a while, when these are like new terms. And so it's a good idea to revisit and see which ones of the list, which ones do you not remember, and then make sure that you can refresh your memory on that. Another question, how about here? When a pupil constricts, so the opening in your eye, when it constricts, gets smaller in response to a bright light, what is this reaction called out of the choices that you have? Negative feedback, positive feedback, osmotic pressure, catabolism, and anabolism. Okay, I'm gonna close it. 5, 4, 3, 2, 1. 89% good, negative feedback. Okay. So that when the light is shining in your eye, the response is to close the eye pupil to lower the amount of light. So it's the opposite of the initiating thing. And I use that term because I have another question for you. So when the pupil constricts in response to the bright light, same situation, what is the bright light called? The receptor, effector, control system, or stimulus? All right, I'm going to close it. Everybody's answered. 5, 4, 3, 2, 1. All right, so we have some mix. This is a little bit more of the nitty-gritty material here. Oops, oh, I didn't put a box in. Oh, man. Let's see if it gives me mine. All right, it gives me my thing back. Well, I didn't put a box in here. So the light coming into the eye is the stimulus. It is stimulating the body. So that could be a smell, it could be pressure, it could be someone hitting you with something, it could be a hormone or a drug that you've ingested. There's some sort of stimulus. That's what the light is. All the other things have to do with stuff that your body manages. The receptor is going to be something that is sensing the stimulus. The control system is either the nervous system or the endocrine system. Those are the only two control systems that we have. And whatever the response is in the end is going to be the effector. That would be like the muscle in the pupillary constricte, which is constricting down. Okay, so these are all parts of that homeostatic mechanism. Okay, so now for today. So that is not my opening slide. I think I might have opened the wrong. That's fine, we'll just go with it. All right, that is not what I want to talk about right now. I apologize. We're gonna start with this one. All right, do you remember we talked about, do I have those slides and y'all, the ones that you have uploaded to? Those slides in the wrong place? Yes, oh darn. That's cool. We're gonna start on this one, protein synthesis. Do you remember what protein synthesis is? Can someone just give me a little synopsis about what when I say the word protein synthesis or the phrase, what do you think of? And you can play in at home too if you want to. Okay, we got enzymes. Okay, then we have transcription and translation. Okay, all are good. All are good answers, because protein synthesis would be the process that would occur to make enzymes. All right, and that process has a couple different steps. Transcription and translation are the two major steps. So great. So I was just trying to take you back to protein synthesis because we kind of talked about proteins. We talked about amino acids forming peptide bonds and making chains. And that's what this all fancy material that you have up here is saying. And it's going to come from DNA being essentially copied by and making messenger RNA, making some RNA that is going to be the map for making different proteins. So that's what's going to happen in the nucleus. But then that map has to get to the cytosol so that it can be read by ribosomes, and proteins can actually be made according to essentially the directions that have been copied from the DNA. Now, the DNA structure we already went over, but we didn't really talk about genes. So a gene is just a DNA, part of the DNA molecule that is going to make one RNA molecule. Okay. And so you could think of the RNA molecule as like a recipe to make a protein, some sort of protein. And so that we call those things genes. And so if there is an alteration in a gene, it's abnormal because of UV radiation or a virus or something comes into the cell and alters genes that are supposed to be the right maps and makes them wrong, then the cell will end up making wrong proteins and will be abnormal proteins. And the proteins are the way that the cell actually exerts function in the body. And so usually those proteins will be caught by the immune system because the cell has to display those proteins on the outside of the cell membrane, little pieces of it. And the immune system says, that's not right, this cell needs to be destroyed. But sometimes they persist, and then we have bigger problems like cancer. All right, so to take you back to the protein synthesis, we have to go back to DNA. And we talked about DNA, and we talked about it having the deoxyribose sugar and a phosphate group. Then we talked about the base pairs that connected the two strands of DNA in this double helix here. Okay. And so essentially the genes are the recipes for the proteins, which is basically to make one RNA. And your book talks mainly about coding RNA, which is going to determine what kind of protein, the structure of proteins that are going to be made. There is some non-coding RNA that is going to support this whole process, but it's not going to be essentially the recipes or the maps for making proteins. And we'll talk about them. We talked about them a little bit, and I'll remind you of them. And then these proteins in the end product, just like was suggested here by your classmate, are going to turn into proteins that either make up the cell, the structure of it, or help the function like enzymes of the cell. So this is super, super important that you get these concepts down. So I spliced this picture, I cut out off the rest of it, that I wanted to zoom in on the different types of RNA. And we talked about the different types, right? We talked about there being messenger RNA, which I think of messenger as like that's the map, the copy of the DNA gene that is going to make the protein. So that's the recipe for the protein, messenger RNA. But then you have other RNA molecules that we've already talked about, but they play a role here, and I'm trying to remind you of them. Ribosomal RNA, that's associated with the ribosome, and then also transfer RNA, which is going to bring the amino acids needed to the ribosome so that they can go in the right pattern. And so here you can see in this picture here the transfer RNA hooked up to an amino acid in which it was finding, and it found it, and then it comes over and it matches up with the base pair that's on that messenger RNA in the right sequence. And you can see up here, you can see this peptide that is bonding with other peptides here and making the amino acid chain. So that's essentially all important anatomic, even though they're really small, important anatomic structures that are doing their job. And this is happening in the nucleus or in the cytosol. This RNA, this messenger RNA being read by the ribosome and the transfer RNA bringing the amino acids. Where is that occurring at? I'll give you two choices. One is the nucleus and the other one's the cytosol. You got a 50-50 shot. Are you all trying to write down what I say? Is that what? Yeah. Oh, okay. I feel bad. You all have to write. You don't have to write. I was asking this process of making the peptide bond, the peptide chains, amino acids here. So here we have the ribosome, and it's reading the messenger RNA, and it's making the amino acids. Where is this happening? In the nucleus or in the cytosol? Okay, we have one nucleus. Everybody agree with her, or anybody have a different answer? You don't get away just because someone talks. You have to actually say what you think. You think cytosol? All right, we got one vote for each, so how about the rest of you? What are you weighing in on? Okay, you remember that vaguely. How about you? You don't have a vote? Avoiding eye contact on purpose? What do you think, cytosol or nucleus? Okay, so we only have 3 votes for cytosol. It is the cytosol. But I thank you for speaking up. Brave person. So all of these amino acids floating around here are amino acids that are just floating around in the cytosol. And so the messenger RNA, it was made in the nucleus, but then it goes through the nuclear pores and comes out into the cytosol where it's going to bind to a ribosome. So the ribosomes are in the cytosol. Okay, so this is the part, the beginning part, where the messenger RNA gets made because it's basically copying the DNA. So the DNA actually kind of unzips, and the messenger RNA's base pairs sort of link up with the DNA in the correct order, and that's how it is going to form the RNA strand. And there actually is an end to the messenger RNA. So it's a little bit more complex than that. So you have parts of the messenger RNA that aren't going to end up being used in the end. So these segments called introns are actually going to be cleaved off. This is a little bit more detail than I want you to know because you'll just forget the cap and the poly A tail. You might know it for other courses, but I don't really need you to know that. But the point is that I do want you to know is that the messenger RNA is going to be made in the nucleus. It's going to by a copy of the DNA, and it's going to be essentially edited or modified from its original form before it leaves the nucleus through the nuclear pores. And then when it comes out through the nuclear pores, it's going to participate in the translation of this message. So this part of making the messenger RNA in the nucleus is actually called transcription. So you're transcribing the message from one gene on a DNA, you're copying it so that that cell can make its proteins right. Okay, is that clear? That's the big picture. And the details don't mean anything if you don't get the big picture. All right, so then after the messenger RNA comes through that pore, so in this image you can see where it all started, the DNA unzips, the RNA gets made, and then it comes through it's small enough that it can come through the pore, and now it's out in the cytosol, and the ribosome says, oh, it's gonna come together, the large unit and small unit, they're gonna come together, and they're going to start reading the messenger RNA. But nothing would happen if these transfer RNA helpers didn't hook up to the amino acids and come over and lock down on the base pairs. So we still are pairing up the pattern on this messenger RNA, and this is the output. This is where the amino acid they're coming together to make the polypeptide chain. All right, and that's how the proteins are made. But that's how they're, just made sort of originally, right? Because we can have a long protein chain, but then you have post-translational processing. So this is an image from a different chapter that we talked about originally. We did biomolecules, we talked about proteins, where you can have a primary protein, right? So it's just a sequence of amino acids. That's what the messenger RNA is making in translation. But then we can fold the proteins, we can twist them, we can put them in forms like a tertiary where they have spot welds. The different parts of the proteins can weld to each other, and then they can even link up with other ones and sometimes in larger proteins that are called quaternary or fourth level proteins. So do you see how this is coming full circle? Some of the terms we talked about earlier are coming back into play now that we're revisiting this in the bigger picture. And so the enzymes that are responsible for helping in this post-translation processing are in the cytosol, okay? And also the endoplasmic reticulum is going to assist, and the Golgi apparatus is going to assist. So when we talked about processing proteins and how the Ruffyar played a role and the Golgi played a role, this is the beginning part that they accept so that they can modify the proteins and make them into their final form so that they can be mature. And generally, a term called a proteome is the whole set of proteins that a cell is responsible for making. And the human proteome is essentially all the proteins your body can make. A body can make thousands of proteins according to your DNA recipe. All right, so I have a question for you here. So a gene is a segment of DNA that contains the code for one blank, which then may be translated into one polypeptide. Okay, go ahead and pick your choice. 5, 4, 3, 2, 1. All right, so we got a good mixture of, so this is a big picture. This is, do you get what's going on in a big picture? So a gene is one part of the DNA, right? It's one part. So that gene is going to be a recipe for one protein. Let's just think of it that way. If the gene is a recipe for one protein that cell is going to make, what else in that list is a recipe for one protein? Most of you got the right answer. So it's RNA. So RNA is that map, right? That map is just a copy of that part of the DNA that makes that protein. And that's commonly referred to as a gene. Is that starting to come down now? Good. I'm recording this. I'll post it on the site so you could rewind to parts that are tricky. If I just want you to get the big picture so that you can start to spin the wheel here. Okay. So now the cells that are just sort of made in your body. They need to make the proteins, more proteins. They need to make organelles. They need to make cytoplasm. Proteins, they need to do their job, whatever that is, depending on the cell that is we're talking about. So why I put this picture in is because not all cells are the same. So this is actually a sheet of epithelial cells, which are tightly bound together, and they all do a job. And underneath, this is a different kind of cells that are in connective tissue. So the cells that are down here, there's a little one right here, and there's one right next to it. These are called basal cells. These cells divide, okay? They divide to make all of the cells in this epithelial sheet. We also commonly refer to them as stem cells because they can turn into the cells in this other sheet. Now, all of these cells are actually the same kind of cell. And so they all do the same function. But do you see they look different? This is a little round cell with a circle in the middle, a darker nucleus. These cells have bigger nuclei. Everything is spread out because the DNA material, which we refer to commonly as chromatin, are unfolded so that they can actively be making proteins. In contrast, this nucleus here is not all unfolded. It's darker and more concentrated. And that's because it is going to be a cell that is not making proteins, but rather a cell that's getting ready to divide and replace the cells up in this area here. The reason why I'm talking about that is because this is more like what real life is like, is that you have cells that are all next to each other, and some of them are making proteins like the process we just talked about, but they might be right next to cells that their main job is to divide into cells that will turn into these. So if a cell is going to undergo division and reproduce itself, it has special stages it goes through. But if it's not if it's just sort of sitting there and growing and doing what it's supposed to do, we call that stage interphase. So a lot of these cells are in interphase, and so they may reproduce, but they're not actively thinking about that right now. They're just making their proteins. All right, and so now I can show you a very dry screen here of these different life cycles of the cell, which all have names. So this phase here, where you have reproduction and cell division, that would be equivalent to like that basal cell, little round cell I showed you. It's going to be concerned with these here. But the other cells, or the other part of the cells that aren't actively dividing, that they're in interphase, and they may still be making organelles and increasing cytoplasm and making other molecules. And those phases are referred to as G1 and G2. Okay, so the cell is just making proteins, making cell membranes. If you remember how the Smoothie R and the Golgi apparatus contributed, and Ruffy R some contributed to the cell membranes by making those phospholipids. But then the S phase is actually not when the cell is splitting apart, but the cell actually does copy its DNA. And so I'm not sure I think I do have a picture in here about the copying of the DNA. So the DNA is going to replicate itself. It's going to unzip, and it's going to replicate itself so that the cell can grow more cytoplasm and organelles, and eventually then be concerned with reproduction for a little while, and then it will go through this phase again, if that's what the cell is destined to do. And we call this interphase cell growth, where it's making DNA, it's making DNA, cytoplasm, and organelles, and then we call it reproduction when it's actively concerned with splitting. Now, and then a lot of the cells in your body, if they're not concerned with growing and reproduction, and they're just undergoing maintenance, they're just making their proteins, but they're not growing more organelles or cytoplasm, we call that G0, which is phase, and those cells are not dividing cells. And there actually are a lot of cells in your body that are not concerned with dividing. And that will become clear later. And so this is just another little schematic here to show you how a cell goes through its cycle of division and growth. Okay, so now I have a question. Within which phase of the cell cycle does DNA replication occur? I just said it, but I do understand how hard it is to stay focused and to know something I just said. So is it the S phase, the G1 phase, the G2 phase, or the G0 phase? All right, take a stab at it. I'm gonna close the poll. 5, 4, 3, 2, 1. So got a couple people don't want to guess. Just throw it out there, it doesn't matter. Take your best shot. No? All right, here we go. So most people put B, which is the G1 phase. Some people put C, G2 next. Okay, so let's go back to this picture here. So the G1 phase, which makes organelles, the S phase, the synthesis phase here, is where we actually duplicate our DNA. So it's going to go to G1 into synthesis, DNA, into G2, or cytoplasm, other molecules, and then through reproduction. And 0 means no growth. Okay, so actually the answer is the S phase. So we're synthesizing new DNA. All right. And so you might wonder, where does this extra cytoplasm and organelles come from? And I just wanted to remind you that we kind of already talked about this, but we were just learning what organelles were and what they did. And I wanted to remind you about the creation and generation of extra phospholipids by making the vesicles that are involved with the ER and involved with the Golgi complex. So those organelles are important for making new membranes, especially membrane-bound organelles inside the cell. Okay. And so the DNA replication on the first parts is going to occur is that the supercoiled DNA is going to uncoil. The strands are going to uncoil and so that they can come apart, because we have to actually make more we have to duplicate the DNA, make a mirror copy so that the cell can divide and end up with the same DNA in both cells. And we don't want to make any mistakes, because if we make a mistake in this, the cell will be abnormal and it could really hurt the body, okay? Because the cell could make bad proteins and it won't do its job well. And that can affect neighboring cells. All right, so now I zoomed into the first one is, we're just showing you how we're uncoiling the DNA, and then the next image showing you the bottom part where we actually have kind of unzipping, and then we have the new base pairs that are in the nucleus kind of just hanging out. They're going to reattach to a new strand on the DNA, and this is going to be done with the help of an enzyme called DNA polymerase, okay? And so what we're going to end up with is a duplication of the DNA. Now, it's not a complete division, and so you end up having connection of the DNA strand in the center, which we'll see in a second. And so there are also segments on the DNA that are called telomeres, and those are the protective endpoints of the DNA, of the chromosome. And so every time that the DNA gets replicated, those telomeres get kind of clipped off, parts of it gets clipped off. And what they used to think is that you only had so many telomeres, and so that they thought that there was a set point and when you would die, essentially, like you would just age out and you couldn't replicate, cells couldn't replicate anymore. But when they found that they actually can be rebuilt by the enzyme telomerase. So luckily, it's not like an end all to cellular division. Yes?
That makes sense. Increase the production of specific. Couldn't you just live forever?
So the question is, if you increase the enzyme telomerase, could you live forever? I'm sure they're trying to do that. I don't know how that research is going and how you would get that into all of your cells. But I'm sure that is an active research avenue for cells that have trouble with division. And there are some cells that really don't divide, like neurons. They don't divide at all. Except for in a couple places in the body, which we'll talk about with the nervous system. So yeah, it's definitely a target of people trying to increase. They used to think you couldn't. They used to think that a telomere just was a set length at birth, and you couldn't actually increase it. But then they found out that wasn't true. All right, and so these duplicated DNA strands are actually still connected to each other at a place in the center called the centromere. And we call these duplicated DNA strands, we call them now, we don't call them chromosomes because they're duplicated into this special configuration. We call them chromatids because they are a duplicated chromosome that's attached to each other. And they're mirror copies, right? That would be like if you had a twin and you were attached to the twin. Not in a harmful way though. All right, so which of the following is not part of a chromosome? All right, make a stab at it. I'm going to close the poll. 5, 4, 3, 2, 1. Okay, so we're pretty much almost evenly divided here. It's cool. That's why I do questions. We learn more questions sometimes than we do from talking. All right, so we have a chromatid pair. So a chromosome, now that the DNA is duplicated, okay, so they are chromatids. So chromatid pair is a part of a chromosome, technically. A centromere is the part of the chromatid pairs where they're connected to each other. So that is a part of it. A telomere is a non-coding part of the DNA that tells when the chromosome starts and stops. So that is part of a chromosome. So RNA was the duplicate map of the gene that is going to leave the nucleus and be read in the cytosol. And that really has to do with protein synthesis and not as much reproduction. So the answer is dRNA. Okay, now that we kind of understand that picture, the big picture is there are two main types of cell reproduction. One is called mitosis, where it's just that a parent cell is going to split into two mirror daughter cells. So the cells are going to be exactly like each other. And then we have another process that's called meiosis, which is where a primitive sex cell or a germ cell is going to undergo reproduction. Yes. It's not. No.
I was always told as a high school.
Oh, the question is it meiosis? You could say it any way you want to.
That's what I was going to say. I was like, they always said in high school, I'm like, wait.
Okay.
I was so
It's all good. You know, people say duodenum, they say duodenum, but we're going to have that happen a lot. No worries. I can't tell you that it's wrong. I can only say that in my training, we say meiosis. But I'm not going to correct anybody saying meiosis, so I apologize for saying that wasn't right. It could be right. All right. And so they also say things differently in different places in the world. So if you're in England, when they pronounce a lot of anatomic terms, it's a very different pronunciation. Different inflection, yeah.
Right.
Yeah. And so meiosis or meiosis is where you actually have a primitive sex cell that's going to turn into a gamete. So you're going to make either a sperm or you're going to make an ova. Okay, so that is a whole different process. It's very important too, because that's the beginning of life. And so if you mess up, if there's an alteration in that reproduction cycle, then you're going to have either an oocyte or a spermatocyte that is going to be altered. All right, and maybe defective depending on the situation. So we're going to talk about mitosis first. It's also known as the M phase. Okay, so the parent cells are going to split into two mirror daughter cells, and it involves a process called cytokinesis. So it's cell movement, where the cell is going to split in half, essentially, and half of the organelles and cytoplasm and nucleus and DNA is going to go one way, and the other half is going to go to another. All right, and the microtubule organizing center is going to play a big role. Hopefully you remember that with the microtubules, because the microtubules is what's going to actually make a spindle and connect to the chromatids and pull them apart, so that as you have these chromatids that have copied each other or attached to each other, one chromatid has to come apart from the other, and each of them have to go into different cells. So the first phase is called prophase, and that is going to be characterized by loss of the nuclear envelope, and the chromatids are going to start to condense, okay, and come together now, and then the centrioles are going to start to move away from each other to opposite sides of the cell. And the spindle that have been, microtubules that have been constructed between them are going to start to come towards the center of the cell. So here you can see it better in the next phase. So that was just sort of beginning. The next phase, you can really see a clear demarcation. It's called metaphase. M for middle, in the middle phase, where all of the chromatids are all lined up here in the middle of this parent cell. And you can see the spindle coming out from the centromeres where it's going to attach to the chromatids. And so it's like, I want one of chromosome 1 and you can have the other one, essentially. And so that when we split, chromosome 1 is going to be both reflected in both of the daughter cells. The next phase is called anaphase, and that's A for apart, where the chromatids are starting to be pulled apart from each other by the spindle fibers that are attached to them. And also, though, although it seems like all of the action is down here with the DNA and the chromosomes, really the half of the organelles are actually going on one side and the other half are going on the other, and cytoplasm and everything. So there's a lot more organization than just the chromosomes. And then the last phase is called telophase, and this is where you have active pinching off of the cell membrane, because we need to have continuity here and have division, complete division of the cells. So we're going to see the cell membrane sort of pinch off and pinch down, and we're going to, again, end telophase with continuity of the cell membrane. All right. So now that we just went through this, I love to throw this at you. All right. So what did I just say? So what is the correct order of the phases for cell division in a non-sex cell? All right. I'm going to close it. 5, 4, 3, 2, 1. 89%. I'll take that number. That's a pretty good number. All right. I think there's a little song I think we've been saying since high school, right? Prophase, metaphase, anaphase, telophase. Anyways, if you haven't, you should probably start singing it. But there we go. So that was mitosis. Now, meiosis is very different, because a sex cell, when you think about it, a sex cell from one parent, from one source, is going to join with a sex cell from another source to make a human cell that is going to have the regular chromosome allotment, which is 46. And we call that the diploid cell. They have 46 chromosomes. And so we have to have the one source of either the ovum or the sperm. One of them or both of them can only have half of those chromosomes. So we start off with a primitive sex cell having 46 chromosomes, but then we undergo DNA duplication, and so you end up having the chromatids that are connected to each other, but then you're going to have them separate and make a daughter cell. And even though the DNA has replicated, the chromatids are not going to come apart. That's one of the changes about it. The other change that you're going to have is that they don't talk about in your textbook, but I would be remiss if I didn't tell you, is that during this process of meiosis, you have crossing over of gene segments, so that you have recombination of the DNA material. So your offspring do not have the same genetic background as you. They have parts of your genes because they cross over, and some come into this cell and then some come into this cell, and you don't know which one got which. And that's why everybody ends up looking different. We don't look exactly like our parents, but we might have some of the traits. All right, and so we end up calling this meiosis. We say that there's two different phases. So meiosis 1 is where you have the chromosomes have themselves in the splitting. So the daughter cells from meiosis 1 make cells that only have 23 chromosomes in them. So we call this the reduction step, the meiosis 1. So we're going from a diploid original cell to a haploid with 23 chromosomes. And then meiosis 2, which is the next phase of splitting, is where you have that haploid cell, just the chromatids come apart and they make a daughter cell they're still not exactly like the parent cell, because as you can see with the colors all mixed in here, that none of these are going to be the same as the original parent cell because of crossing over. You end up getting crossing over between the chromosomes. And so, I thought I fixed this, I guess it didn't. So in the female and the male, this process is different. And that's because in the male, we're making sperm, and sperm is made throughout a male life from puberty to death. For a female, the oogonia, that is actually, a female is born with all of her ova. And so essentially some will regress, and only some will be stimulated to reach maturation and to actually form an oocyte, whereas for the male, most of the developing spermatogonia will end up becoming sperm. And for every one spermatogonia, actually 4 sperm are created. But it's not the same. For every one oogonia that gets stimulated in the female, only 1 oocyte gets created. And so it's not the same process, although they do involve both a reduction step and then a separation of chromatid step. So this is, I'm not going to ask you these details on the next 2 slides. I just want you to understand this, because this is a part that trips up people. And so if you just start off right, then I think that you'll be able to follow this. And some of you might be interested in reproductive health. And so for a male, they have 1 sperm. It's the primary sex cell is called a spermatogonia, and it's going to have 46 chromosomes. And then the cell is going to separate out, okay? And it's going to, and this is meiosis 1, and those chromosomes, although they duplicated, they also crossed over, so they're all new, and they come into make 1 set, comes into each of the splitted cells, or cells that split, but they're not mirror images of each other, and the chromatids that they duplicate are still attached. So it's just that chromosome 1 comes over here, but that chromosome 1 is also attached to its sister chromatid, so they a pair of chromatids, and this cell as well. And then the chromatids just come apart in meiosis 2, and you end up with 4 spermatids that can eventually maturate into sperm. So that's how it's different. And then for the female, there's a primary oocyte. And although it does happen in a similar way, one of the resultant cells from meiosis 1 gets all of the cytoplasm. And so it doesn't share very well with the other cell. And so this cell isn't viable. It doesn't get any of the cytoplasm. We call that a polar body. And then in the second division where the chromatids come apart, it happens again. And so this is the second polar body. And so we only end up having 1 mature ova. Okay. That happens here. All right, so which type of cell division occurs only in primitive sex cells during the process of mature gametes? Take a stab at it. All right, I'm going to close the poll. 5, 4, 3, 2, 1. 86th percentile. Yes, I'm so glad you all are listening to me. Meiosis is one of my favorite topics as I do the reproduction at the last 2 schools I teach at. Okay. And I also, one of my specialties is human development. So I like meiosis. What regulates this? You might be wondering what regulates the cell's life cycle. You might not be, and I get it. I'm not really that interested in it either. But just in case you are, there are some enzymes that are called cyclin-dependent kinases, okay? And a kinase is an enzyme that's going to activate a cascade inside of a cell. And so these kinases are responsible for activating the cell to make it into different life cycles. So they're kind of like proteins that say, okay, now it's time to do this, or now it's time to do that, and go into the different cycles. So different cells have different life cycles, and I alluded to this earlier. So here we have this epithelial sheet here, and we have these cells that are on the bottom. Those are also stem cells or basal cells, and they are destined to undergo replication and replace the cells that are in this layer. So if I had an insult, if you had an insult to a cell that's here in a more superficial layer, that's probably not as bad, right? So this is actually the bladder. And so inside the bladder, we have urine. And what happens if we get injury to one of these cells and it dies? Well, it can be replaced by the stem cell. But if we have overall inhibition of reproduction because, I don't know, the person's undergoing chemotherapy and they're taking drugs that are made and cause the body to decrease mitosis, then we might have more of a problem healing from an injury to this cellular sheet, right? You can see how reproduction and cell growth is actually can be a really big issue that needs to be constantly tended to in the body. And so and this was just a pretty picture of the mitotic spindles, and here you have all the chromosomes lined up here. All right, and so what I also wanted to show you this, let me see if I can get this pointer, this is my new pointer, to work. No, I guess not. It doesn't work the way I want it to. That's okay. All right, I can click with my mouse. So this is actually the virtual embryology site at LSU, and one of my old teachers, he sectioned all of these embryos, and so this is the histology slice through the embryo, and so he made it so that we could learn off of it. And this is the aorta right here. This is the suspended heart that's developing in this embryo. You can see it, how it's sectioned down here. And this is the gonadal ridge. So there are some little tiny cells here. I can't really zoom in much closer in this section. Let's see if I have a step. Yeah, we have more gonadal ridge. So this is the little ridge where all of the sex cells originate in the body before they migrate down to where during testicular and ovarian development will be. So it's right next to the aorta in your lower back region, essentially, of the embryo. And I just thought that was cool. Also, what I wanted to show you, let's see if I can come into something. I wanted to show you, so this is the spinal cord, developing spinal cord right here in this area. And you can see how it's just not very organized. We don't have as many layers, and the organization, the neural tube inside, neural canal inside is very wide. It's not going to be that way in the end. And I wanted to just point out that because the cell divides and starts off in one point, that's a very basic cell that is going to further gain specialty, and we call that process of that cell going to become further differentiated. So we talk about cells and how specific they are in their function, and cells that are more differentiated and they have more highly functioning jobs and they do a lot of things already, they're usually not the cells that are dividing. And the cells that are doing a lot of division are going to be ones that aren't doing a really high amount of functions, like for a particular organ. And that is kind of a general rule of thumb that will continue throughout the remainder of this course. All right, all of the proteins synthesized in the whole body is called the what? All right, I'm going to close the poll. 5, 4, 3, 2, 1. All right, so we're a little split. We're split on a human proteome and on protein synthesis. Okay, so all of the protein synthesized in the whole body is called it's actually the human proteome. So you can have a cell, proteome, the proteins that are in the cell, but the whole body is human proteome. And, the whole body, you can think of the hundreds of thousands of proteins that can be made together. This is a little more of a minor point. It's just a term that they had in the chapter. Okay, so that is the end of the lesson. I forgot to put a slide in here on, there are some terms in the textbook that I did want to go over. Let me see if I can pull that up right here because I forgot to put that slide in here. They are in your objectives and in the lecture study guide for you to look up, but you might not. Here we go. So I do want you to know these terms because they are very important for when you're talking about cells and tissues. And so this is the slide I must have forgotten. And you might want to, I'll add it in there. I'll add this gray box into your PowerPoint presentation when I finish this. And so all of the cell functions that we have talked about today have been normal cell function. We're just talking about a cell that's just trying to live a happy life. It's just trying to make the proteins that it wants to make. It's just trying to replicate and replace cells in the epithelial sheet or do whatever it's supposed to do. Well, if a cell is actually smaller than it's supposed to be and has less function than it's supposed to be, then that's going to be noticeable. So the organ or tissue is going to be a lot smaller and it's going to have a lot less function. And we would call those cells atrophic, or we're going to say that those cells have atrophied. So that's an important word that you should be able to refer to cells. So atrophy means that the cell has less function and less size and structure. And on the flip side, if the cell is larger and maybe secreting more than it's supposed to, it's overstimulated or for some reason overproducing something, a protein, we say that those cells are hypertrophied. So those are opposites. And then we have a term that means having cells that have increased in number. So you can have cells that increase in size and we call that hypertrophy, but if they increase in number, we're gonna call that hyperplasia. And then if a cell changes its shape or size or the type of cell that it is in response to some sort of stressor, we call that dysplasia. And that happens a lot, like for example, cigarette smoke in the respiratory epithelium, so in the bronchi, the epithelium will change from its regular ciliated pseudostratified epithelium to a stratified squamous epithelium. And we would call that dysplasia because it's not supposed to be stratified squamous, but the body is doing that on purpose to try to protect itself from the cigarette smoke. And so that's one example of how cells can change their shape because they want to have a different function. They want to increase their protection, and they weren't as protective with the little cilia sticking up. All right, and then the last concept I wanted to talk to you about for that is that a cell can undergo regular programmed cellular death. There are times, especially during human development, where an embryo will actually overgrow. So the certain tissues will proliferate a lot and it will fill up a space. And then, especially if you have a tube, that space needs to have a hole in the middle. So the cells in the center of, like, for example, your esophagus will undergo something called apoptosis, and that's regular programmed cell death. So it doesn't cause any inflammation in the body. It's not an actual injury. It's what's supposed to happen. Otherwise, you wouldn't be able to swallow, or an infant wouldn't be able to swallow milk when it was born if it didn't have a recanalize its esophagus. And so that's a regular good process that doesn't cause any inflammation in the body. On the flip side, if you have a stress or an injury that damages cells, so they lose their function, they release their contents, and they die because of stress, that's called necrosis. And that process does actually simulate a inflammatory response by the body, and it can damage all the cells around it, and it can decrease the organ's intended function. So those are terms I do want you to know for this session, and this concludes the new material for this first test that will be not on Thursday, but on Tuesday. And so on Thursday, when you come in, we're gonna do rapid fire review session, where we're gonna do a lot of turning point questions and see what it is that you're still having trouble putting together. Okay, does anybody have any questions? It's all good? Really? You did it in genetics, okay. Did I do a good job? Like, yeah, did I say anything wrong? You'll let me know, right?
He gave us the very detailed version, like
Okay.
beta and alpha and DNA ligase.
Yeah, you don't need to know that from me. I'm glad you do, though. But you don't have to. I mean, I try to teach it to where you don't have to have previous knowledge of where we start, because some people didn't take genetics. I've never taken genetics. So,
Easy.
yeah, it wasn't easy.
Don't take it. Yeah, don't take it.
All right, well, you can probably handle it now. Well, thank you all for joining us. Let me know if you have any questions. Otherwise, I will see you on Thursday.